Okay, thank you. Thank you so much for giving me the opportunity to present to you. Blooming Minds online classes. I would like to share my screen. So, hope everyone can see my screen. I'm Nishen from Nishi, Sri Lanka. So we are doing a translation bioinformatics program. We for this test session, we have been requested to conduct a basic NGS analysis training program, introduction training program by Bioscience Online Classes India. So for the day, I would like to present the very basic theory of NGS basics and what is SRA files and what's the difference between FASTA and FASTQ file and FASTQC analysis and what are the pipelines usually generally known as the sequence data analysis of transcriptomic analysis and in terms of metagenomics analysis and what is the output dump data and what you can do with that. Okay, so before we start, I'm not going to give you a basic idea of NGS. NGS is a next generation sequencing. Yeah, sorry. Okay, good afternoon. Yeah, good afternoon. Yes, so NGS is next generation sequencing. So in this whole genome sequence analysis, right, if you take about human genome projects in 1990s, right, so the one sequencing, the cost of one sequence, it was around 2.7 US dollars, 2.7 US dollar billion. And the techniques was very old and it's known as Sanger sequencing. And later on the technological development, so there are many commercial sequencing analysis machines, and that has brought down the sequencing cost drastically from 1.5 million or 100,000 to 10,000, and now 2015 it's 5,000, and if we talk 2020 sample analysis, it will be less than $1,000, right? And actually we can optimize less than $500, but if we just concentrate the in enzymes and the utilities cost, right? So exclusive of the handlers expenses. But apart from this, okay, So from the starting, I would like to see the audiences. So there are many students from undergraduate, I guess, and some of them are doing masters and PhD. That's the information I have received, right? So I would like to know if anyone have done NGS or bioinformatics data in a serious research project. So maybe it might be boring for you since it's a very basic session. So before we talk about all the NGS data analysis, right? So where do we get this data, right? So well, let's say, because everyone talks about data mining and data science in this 20th century. So in this day, bioinformatics is one of the biggest data influencer in terms of data sciences. So where we can access this data is one of the data bank is NCBI, right? So there is a Japanese data bank and European EBI data banks, right? So this is NCBI is for National Center for Biotechnology Information. Is from a US based data bank. So in this compared to any other data sciences, right? They have majorly they have a bioinformatics, right? Almost all of this data are from bioinformatics background, right? So from this, right? Let's say we are going to do a basic analysis, data analysis or next generation sequence file, right? So how do we obtain this? So to obtain this, right, we have to use this link NCBI data analysis, right? And you can sign in, or without signing also you can work with this. So there are particularly different data banks, for example, gene data banks, protein data bank, or sequence analysis, or any other types of data banks, right? So here you have to go to ncbi.nlm.nih. From here, you can choose SRA data information. Okay, so this is the SRA sequence read archive. So sequence read archive, so known as sequence read archive, and from here onwards you can get the basic up-to-date data information which has been publicly published. Okay. So in this case, right, in case let's say everyone the current trend is coronavirus, SARS coronavirus 2, right? So let's say I'll just say the SARS So let's say SARS2. Okay. So for example, if you search this, you may get the data files of this. And if you want to particularly choose one of the data file, so you have, what you have to do is you need to choose the data files, and every single accession number what you get here is based on one prepared sequence analysis file, right? So one run. Okay, and you can customize the search settings here, like for example whether all of the research, right, out of 22,242, I think is a public access. Any of you can files, right, without, you don't have to pay or you don't have to do anything, but when you publish the data you need to get the consent of the author, right? And out of this 22,242, right, there are DNA source and RNA source, and there are 2,700 genomes, right? So by clicking this, you can choose the types of data single accession. And there is important things to consider is the library layout, right, and the organism here, right? So if it is an organism, right, definitely this is a SARS coronavirus 2, right? So you can get a random all coronavirus analysis, so single and pair in the sense there will be a 5' to 3', and it's only a mRNA. So that's a single and paired sequence analysis, right? So whether it's a sequence has been done based on both the strands or single strand, right? Single active strand, right? So otherwise you can get platform, right? BGI-seq or Illumina or Iron Torrent or Oxford Nanopore. So depending on what type of the sequence you want, you can choose and narrow down your searches, right? And you can choose for BAM and FASTQ, So BAM is a binary alignment file. So FASTQ is a FASTQ file, and where we are going to get this information. And there are aligned data also. So let's stick to FASTQ. And since SARS coronavirus, I know it's RNA based, so then let's choose RNA. And the best paired sequence are better, so I'll just choosing the paired sequences, right? So then you can get a sequence, whole genome sequencing of this file. So by right clicking the whole genome sequence, right, I'll get an accession number for every single sequence here. So this is an SRA data, and if you want to get more information on particular this file, right, so this every single accession ID tells us that you have to take particular information on that, right? So if you want to know more about these particular researchers, you can click the choose show abstract, and you can go to this PRJNA number, which is a research project number, research project accession number. By clicking that, you can reach the research center which is publishing this data, right? So by clicking this accession number, you can come to the severe acute respiratory syndrome coronavirus 2, and who is doing this CZB Cilia Hub Consortium, CZ Biohub, UCSF. So it's one of the leading scientific institutions in the world, and they have done so far 888 SRA experiments, 178 nucleotide NGS data, and 2136 protein sequences, and totally they have 883 biosamples. Okay, but apart from that, right, what are the other information we can know is, right, so this is the SRA experiment data, right? So we can see the analysis, so the basic information and where this has been done, then the sample information. So in the sample information, we will read this. I mean, there will be huge, right? I think 5,694,000 lines, right? So it's huge data set, right? So we can't. It will take a lot of time to download this data. Okay. Sorry. Yeah. Okay. Thank you. Okay. So so in this session, right, so let's say if you want to download this data, right, so what we should do is we can get the files. I mean, what you have to do is we can go to the metadata. So this is the XRA information. So to download this file, right, what you have to do is download the fastqc dump command, right? So the tool fastqc dump, you can download it from internet and directly download it using fastqc dump command. But if you are a Windows user and if you would like to download this file, you can click the download fastqc, and I have opened this file, right? So then what I have to do is from here I have to copy and paste the srx file name, copy and paste it here and show the runs. So once you show the run, automatically it will show you this is the file, right? It is 16.6 GB file, right? And what you have to do is you have to choose whether you want a FASTA file or FASTQ file, right? You can filter or clipped file also, you can download it, but you can choose whether you want a FASTQ file or FASTA file. And all you have to do is click the download button, and automatically you will get the downloading file. I'm not going to download this, right? Because I have already downloaded this. Okay, so what's the difference between FASTA and FASTQ? Okay, can I ask the questions to all the students here? Anyone? A basic difference of FASTA, or known as FASTA and FASTQ?
FASTQ contains the quality sequence also.
Yes. Okay, this is Prateek. Okay, thank you for the answer. Yes, FASTQ contains the quality sequences. For example, right? So this is the sample file I have already downloaded, which is very small file compared to the file we have just seen down, right? So let's say I open this file, right? So when I download this, I'm sorry, okay, when I download this file, it will be like sra data 1 fastq.gz. It's a gunzip file. So what I have to do is I have to unzip this, and I will get sra data 1 fastq, right? I can open this file using notepad, right? So once I open this notepad, right, if you see this, it's slightly different. I mean, this fastq is each and every commercial application, they have a different sign to start, right? Most of the common signs are greater than sign, right? So usually it starts with greater than sign, but in this machine applied by a system, they have starting with at sign, right? So all you have to do is, right, you need to know what is the quality data here and what's the sequence data. So in this case, there are 4 lines and 2 header lines, right, and 2 data lines. So the header line says very simple, starting with an at, right? Because of this machine, it shows an starting with at srr number and the line numbers and the total length sequence. So which is, this sequence is 74. Okay, and at the same time, right, since this is a fastq file, right, each of this data, I mean each of the base pair, it will be denoted as a quality length, right? Quality length denotes means, the quality length means this 3 a f or g, right, means particularly this g means a's quality, okay, and this question mark is a symbol for the quality standard of C, and this A has a quality of pre. So for example, if I choose this A and this A, right, they are in a different quality, right? So in appropriately, if I talk about this, A is higher quality than 3, because usually it starts from 0 to 9, then A to Z and other symbols. So this has a better quality. So the chance of probability being this A is A, right, is the quality standard of A, and this C has a quality standard of question marks, which is really high chance of C, right? For example, if I compare this G, C, it has a quality standard of G, which means 0 to 9 and after A, B, C, D, E, F, G, right? But whereas this C is 0 to 9 and A to Z and question mark. So it's more higher quality than this, right? So how do I check through this, right? How do we verify before we start the NGS, right? Most of the students, what they do is they will just download the FASTQ files and they jump into the sequence analysis, right? Even before they testing whether the sequence is accurate enough or not, right? So for that purpose, right, what you can do is you can download, right? You can download FastQC program, so which is from Barbraham Bioinformatics, right? It's a FastQ quality checking program, right? So if you download this, you have to download this, and it needs a Java runtime environment as well. So downloading this program is very simple, and you don't need any installation. So once you download this, right, what you have to do is you have to create the FastQC program, and it has a run fastq.bat. This is the executing files, right? Right. So automatically, once you download this FastQC, so this is the program. Just checking the messages. Yeah. Okay. Thank you, Sarathi. Okay. So FastQC files. Once you open this, so this is a sample file I have already downloaded. I'm going to choose the sample file, and it's a gunzip file, but you can use this one even without updating it, right? Or otherwise, what you have to do is you can either extract this, or after extraction with FastQ file also you can import it, right? So it will work in both ways. So once you open this, right? What you see is a FastQC program, right? And it will tell you the basic sequence analysis. So automatically this will say the encoding is used as Illumina 1.9. So that's why we have got this code, right? The at sign and the plus sign, right? So these are all the plus signs denotes the quality standards, and the at sign header is based on the sequence arrival. So this is a sequence. So header after the every line after the header signs from at is a sequence data, and every header after plus, right, which is a quality file, right? So that is the basic difference between the FastQ and FastA, right? In the FastA, what happens is you don't have these two lines. I mean, this will not be there, right? Instead of that, only the sequence read file will be there, right? FastQ. I mean, FastR, you have only this information. There won't be any quality standards. But in case of FastQ, right, it has both the quality data and the sequence data, right? So I'm just okay. So I have gone back. So after doing the FastQC analysis, so this is the basic information of FastQ. So you can see the data file name, conventional basis, and the what type of encoding, Sango Illumina 1.9, and the stored sequence and the sequence length and the GC content. So from this you can consider whether to go ahead with this data or not, right? So the GC content of this total GC percentage is 64, that which is quite normal, right? So we can see, right? But that's not the only parameter we are going to check. So we are going to check many others. So this is one of the important thing. In this data we have position read and the total quality standard, right? So the quality standards above 38 is not necessary, so it is not mentioned. But in this one, total position read, if we read about this article, right, less than 28 base pairs is mentioned. I mean, above 28 is mentioned as green, in between 28 to 20 is orange, and less than 20 it's mentioned as red color. So which means whatever comes in this has a very low quality reads, right? Compared to orange is in this area, it's a medium quality reads, and the high quality reads are on green, right? So which means high quality means in probability, statistical probability, high quality means you can consider those data as an accurate data. So for example, if you consider this okay, for example, if you consider this quality standard, right, for example, the C at T, right, or example this A and this A, right? So which is one is G and one is C, so which means two different type of quality standard, right? So which we can just judge as two different qualities, right? So the more the quality, the accuracy will be higher, right? So which means A adenine becomes A, right? O adenine being A or something else, right? Or maybe it can be T or G or C, right? Or either another pyripurine or pyrimidine. Okay, so this is the quality standards of that, right? So in case if we have to do an analysis based on this, what we have to do is we have to trim this data and remove this data from the data FASTQ file, and we can do the analysis based on only the green section, or maybe we can take orange depending on the data we have, right? Then we have a per sequence quality score, right? So usually the average quality score, which is higher, 36, 37, which is okay in this case. Then we have to consider the per base sequence content, right? So the per base sequence content, right, which is not common at all, right? Why? Because it has a two different standard, right? So T and G is too high in the beginning, but there is a moments here. So we have a problem here, but again, if we check this one, I mean, actually we can consider sometime, but if we try to consider the GC content here, the GC content is totally the different results, right? So GC content, we check through GC content because there are two important reasons. One is they have G and C has a more number of hydrogen bonds, right? So the attachment is higher, and every organism in this world, they have a particular unique GC content percentage, and that percentage you can do a normalized standardized curve. And if you do a theoretical distribution of the blue line depicted as here, right, which is not aligned with the actual DC content per read based on the sequence. So in this case, right, we can conclude that the data file what we have downloaded is not accurate enough to move forward, right? So what we can do is we have to remove the unnecessary low quality data, right? And then we can run this FASTQ again and see whether the GC content improves, right? If that is the case, we can go ahead, or otherwise this doesn't, I mean, this is not, I mean, this file is not suitable enough to carry on, right? And the per nitrogen base is fine, and duplicates and the adapter sequences. So overrepresented sequences and adapter sequences. So these are other two values from FASTQ format, right? Where, because usually the sample preparation of NGS data analysis, I mean NGS sequencing needs adapters. Similarly, PCR primers in PCR, right? They need adapters to replicate and mark it. So these adapter sequences can play a role like primer dimers in the basics. So to remove these, right, usually the most commercial adapter sequences will be recorded and it will be identified using this. So here there is a no hit, but there is an adapter sequence 191, right? At the same time, so this is an overrepresented sequence, and there is an adapter sequence. So it will, the adapter sequences from Illumina universal adapters or Illumina small RNAs or the Nextra or solid or Illumina small, right? So this, if there are adapter sequences mixed with this data, it will be automatically identified here. Okay. So then once we get this one, and once we have a better quality standard, I mean, to get a best quality standard, right, you can see the file. I mean, this is the program for Abraham Bioinformatics, right? So you can see how a good Illumina data will look like. So usually a good Illumina data will look like per base sequence quality should look like this, right? And GC content should look like this. So this is, well, I mean, you can't get a superimpose into theoretical distribution, but still GC content is almost okay, right? And the overrepresented sequences or adapter sequence, it should be 0, and the end content, right, per base sequence quality it should look like this, right? At the same time, the bad quality data, okay, so if it is a bad quality data, usually look like this, and majorly above 26, 24 to 26 larger fragments are in a very low quality. So in this case, we can't trust the sequence quality here. At the same time, GC content is too different, right? Not superimposing, and the length distribution and the adaptive sequence is fine, but overrepresented sequences, right? It's similar to primer dimers, so overrepresented sequences are there, so which means it's not good in quality, right? So that's the issue here. Okay. Give me 1 minute break, guys, please. Okay, thank you. So a small technical difficulties. Okay, so fine. After the adapter sequence, so once we okay, I'm audible, right? Okay, so once we decided whether we have to go ahead with the sequence file, right? The next one is the pipeline analysis, right? So we need to know what kind of pipeline we are going to go ahead, right? So let's say, for example, in Illumina data, right? Sorry, Illumina or any SRA file, right? So there are different types of data analysis, right? For example, transcriptomics or metagenomics. So it can be whole genome sequencing, it can be whole exome sequencing, whole transcriptome sequencing, and metagenomics or metatranscriptomics. So in this case, each of this file, it should be, I mean, you have to, before we start, we need to know what type of the file is, right? So if we get a file on transcriptomics, we can do any metagenomic analysis, or we can do any exome, whole exome sequencing or whole genome file. At the same wise, likewise, whole genome file, we can do any transcriptomics analysis. So each of these data analysis file, I mean the library preparation, is particular for that particular type of analysis, or in the other way, the particular type of analysis should be made in terms of the library preparation, right? So let's say we have a library preparation of a TGCA, the whole cancer library, oncology research projects. They are doing most of the transcriptomics analysis. So if we have a file like that, right, then we need to do a sequence or series of different types of analysis, right? It's not a one particular program which we are going to do the analysis. So from the beginning, right, from start, okay, so from the start to the end, right, there are many other program you can use, and each of these tools, right, these are all online tools. Most of the time it's online tool, but there are server based tools as well, right? For example, we are partnered with PineBio US, so you can use their online server system, or uh, or else you can download your own software tools to your Linux platform, and you can have your own servers. Or there is a Galaxy projects, or similar to, there are other online tools, right? But if you see, um, each of these programs are very particular, and you need to know what happens in each of these program before you start the, or before you plan your analysis, right? There is no proper correct way, right? I mean, which means in terms of conventional law that you should do this kind of analysis. But there are types of analysis. For example, there are several tools for pre-processing, several tools for data simulation, several tools for error corrections, mapping on genomes, mapping on junctions, or exon detections, or alignments, or isoform construction and GTF processing, mapping on transcripts, expression level, differential expression, segmentation, annotation, and end. Right. So there are many types of tools, and you have to decide what tool you are going to use. And each of these tool, depending on particular data type, and there is an advantage and disadvantage for every tool, you name it, right? So you have to choose, right, a particular tool based on why you need this. Explained because it's a very small presentation, so I have a time limitation to finish it within 1 hour, right? But even though, right, so I would like to give you a very brief information on how you can plan this, for example, right? So I can't give, I mean, in case if you want to learn this NGS analysis, let's say, for example, bow tie or for exomes, transcriptomes, or metagenomics analysis. But let's stick to the basic session today, right? So for example, pre-processing. So pre-processing in the beginning of the analysis. So beginning of the analysis, there are different tools. One is Clean Primer, Array to Faster, or PCR Clean, Tremomatic, or JTF Adjust. So each of these tool has different function. So for example, I have shown you this particular data, right? So if I have a data like this, right, I should know what I should do and what is the next step. So in this case, right, there are good quality data and bad quality data, and there are basic no adapter sequences, but there are overrepresented sequences. So I should do, right, I have to remove these overrepresented sequences, as well as the per base sequence quality. I need to moderate this. So which means I have to remove the low quality sequence first of all, and to make the file better. So in this case, right, I have to use Trimomatic, which is a tool designed for to making the to have a better quality data, right? So trimming of the low quality data, and the PCR clean will do a better performance, right, in terms of removing the adapters and overrepresented data, right? So in that case, the quality data what we have, I mean, most of these data which in this file, right, some of the fragments will be removed, right, because they are in a low quality. For example, if this file is fragment is very low in quality, right, this will be removed. And if this quality is very low, and that will be removed, right? So likewise, okay. And in case if you want to read how many numbers of this each line of this data I have, this is 1.1.1, right? So just you open up this, it has the 3183. So 3183 total sequences they have. Okay, so these are the basic data. So once we remove this data, the low quality data and overrepresented data, so adapter sequence, or any basic cleaning, so that is why this is known as a pre-processing, right? Because we didn't even start doing any NGS work, we are just making the file, the sequence file, better. Okay, after that, right, we can go for a mapping on genome, or mapping on junctions, or exon detections, or this fragment analysis. So there are different type of this. One is bowtie, top hat 2, high set 1, high set 2, and bowtie 1 and bowtie 2. Okay, and there are star and B, right? There are different tools, right? If you want, you can use those particular tools, right? So let's say if you have a transcriptomic data, and each of these bowtie, top hat, and high set have a pros and cons, and you have to choose what you really need, right? So, and each of these for analysis, we need a reference alignment, right? So mapping on genome or mapping on junctions, so where this sequences has been done. So for that, each of these sequences will be mapped to human genome, right? The latest human genome fragment, right? And it will give a GTF processing or ISO file processing basics, right? So what happens is these fragments will be mapped with the existing GRCH G38 sequence file. So from that point onwards, you will get a pre-processing numbers for that, right? So after this high set or top hat or what I do this, right, then we will know, right, if it is a transcriptomic file, there is an additional file. I mean, if there is a whole genome file, this will directly lead the isoform preparation. But if it is a transcriptomic file, you need a cufflinks and cuff merge file. Why? Because how many times this particular sequence or data is represented, right? So you need to measure those information. So to measure that, you need additional cufflinks and cuff merge, right? So most of the time you might be wondering why bow tie and cufflinks and cuff merges are there, because they have named it like that. Like most of the jewels they use. So then after the merging, right, you will have an isoform construction, and the mapping on transcript will happen after cuff merge. Mapping on transcriptomics, both I will do, right? So once we do the RSEM exponential tables, right, your data will look like okay. So for example, this FPKA, right? So you will get multiple data here. Some are CSV files, some are read counts. right? So we can get FPKM, right? It will be TPM, FPKM, RPKM. So if it is a transcriptomic file, right? Let's say you can give multiple SRA files, and if you have a multiple SRA file, you will get this data, right? I think this is better if I open it through a CSV file or maybe an Excel file. Okay, so if I open it through Excel file, I have around totally four samples, right? And total four samples, the information, right? So these are the additional numbers. So you can ignore these columns, right? So this is a working file. So for example, if once the analysis is done, right, analysis will show you, right, how many types of FPKM or TPM figures, right? So this is FPKM in CSV file, and based on, right, individually four different samples, which means four different SRA data file, and out of the SRA data file, right, after the GTS formats, right, first they will find each fragment they align with ENSG number, right? And if you are wondering what is this ENSG number, right, what you should do is um, yeah, so this ESNG number you can search it from this, and ensemble you can get the gene identification number. So I'm sorry. So this each of the file has a gene, and you can get the basic information of that gene and where this is located in human, right? So 8q22.2, right? By ensemble means you can locate your chromosome and the position of that, which will be replicated by a red line, right? And in case if you want to read more information, you can go to UCSC genome browser and do a basic analysis, or otherwise the basic information on this is more than enough. Okay, and so, and if it is a 0 or less than 0 means, that means all the data that has been detected, these were 0. So for example, the first, all of these I have no information, so which means it has not detected any data from this, right? Especially because of this is a particular type of transcriptomic file, and because of that not all the transcriptome, right? I mean, if it is a tissue specific transcriptomic file, all not all the data will be expressed by all the tissues. So it will be some tissues doesn't express the data, right? So in terms of, we find out for different types of data, you might get 0 or a very huge differences in between this, right? So for such as this, so this is one of one type, particular type, whereas two sample test, they are from a different type of patient. It has been expressed 1.00114 and 2.0, right? So we can see that is a differentially expressed two different data sets. Okay, so if you want, you can search that as well, and what is this particular gene and how this has been differentially expressed. And if you want to do more analysis based on this particular sample file, right, what you can do is there is a, differential expression, differential sequence expression. So what happens is you can upload this data, right? Based on this, right, you need two different program. One is DSeq, and the second one is the KEC pathway mapping, which is designed for DSeq. didn't, I don't want to do it now, right? But if you have both of these dseq and kick pathway files in your laptop, this file is enough to run the kick pathway on the dseq, and which will produce you similar data files, data images actually. So for example, this is, I'm sorry, these are data sets. Kick pathway files. Okay, give me a second. I'll just show you a basic file. So this is just downloading some random sequence file. This is a path view file, and another file. Okay. So this is a path view file. So this is done by kick pathway and on DSEC, local computer or server, both will give you the same results. So in this one, ECM receptor interaction. So based on the Excel sheet, what you have been got, right, the different for the whole genome on different 4 different patients. So in this case, S1, S1 and S2, group 2, S1 and S1. So there are 2 groups on differential expression data. So based on that, this has been generated. Okay. So if you download this and refer, this is a different one, right? For a factory transduction, right? So automatically this will give you draw it on the data sets, right? If you have, if you, I mean, if you have used Keck pathways for biochemical studies, biochemical history analysis or studies, it will be easier for you. So, and you might know what this is. So the basic plasma membrane, and it will automatically show the particular gene responsible for this particular protein synthesis or the action, and it will show you what is the upregulation and downregulation. So -1 is downregulation and +1 is upregulation, and so the red here is upregulated. So in this case, particular ODOR detection, R is upregulated, PKG is upregulated, and Golf GOLF is downregulated. In this case, R and GRK are upregulated and GRK downregulated. So the red colors are upregulated and downregulated. So this is different data sets in terms of Alzheimer's disease patient. I think, yeah, this is a DEC HSC. Yeah. So this is a patient sample of 2 Alzheimer's patients. So then if you see one sample based on two groups, whether control groups versus the Alzheimer patients, there are differential expressions. So which means based on the control groups, I mean according to the control group, the patients who has Alzheimer's, they have a huge difference in terms of higher upregulation, right, in major sequence upregulation in ECM receptor interactions, right? So if you see this ECM versus Zintin, right, I mean, this is not the only data. I mean, there are many data. So I'm just showing you a basic data set analysis, right? So this will be the end product in terms of data analytical sequence using Python or built-in tools like DSeq or differential expression tools or heat maps, many more bioinformatics tools. Right, so this is one example of one interactive examples of what we are doing. Okay, and in terms of metagenomics, you can do many other data plots or hierarchical identifications or OTU preparations. In whole genome sequence and whole exome sequences, right, you can find the promoter sequence data or annotations on the whole exomes and whole genome sequences, right, and there are overrepresented analysis as well, right, and you can find out SNPs, indels analysis or proven analysis of proteins, where you can find out whether this protein that has been utilized, I mean the perform, I mean the synthesis, I mean the mRNA produced proteins, whether they will actually work or not, right, based on the IQ. And there is a isoelectric point analysis. You can find out whether these proteins are actually functioning proteins or it will be too much for a day. I understand, right? So just wanted to know any questions so far. Yeah, I can understand that this is within 1 hour. It's really too much. I mean, usually this analysis would take 1 day or more than 1 day, 1 to 2 days. Usually it might take sometimes 2 3 days as well, right? So to teach you all this analysis in 1 hour is not practically possible, right? But still, I hope you might have learned something little at least. And I would like to ask some basic questions since I have a time duration of 1 hour, right? So if you have any questions, I can just answer the questions. Okay, there was one question. One question from Pratik Paul, right? Any open source or free software alternatives? Most, I mean, the software I have, I mean, the visualization, the pipelines was a paid one, yes, true. But those are the GUI is paid versions, but all these softwares are free and open source, right? For example, you can download all of these softwares, all of these Bowtie bioinformatics tool. So all of these tools you can download it through online, right? Bowtie bio source course, right? And these are not Windows GUI program. You need open source. I mean Linux, Ubuntu Linux, or any other similar Linux, Ubuntu Linux server. You can automatically download, right? For example, if you are using command prompt, I mean the terminal in Linux, right? I mean I'll close this down. This, I mean, I can't show, I mean, this is not appropriate. I mean, I can't use this as an alternative of Linux, but let's say if you have a terminal, right, it's like sudo aptk, you have to download like sudo apt install. Go type, right? So automatically if you download, I mean, if you install using this, this is enough, right? To install this, you need an active internet connection and this, right? But this doesn't work in Windows, right? So these tools doesn't work in Windows, so you need an Linux environment. And all of these tools are free and open source. You can use it. They want, and most of the research level, you need to have your own research. And yeah, you can use cybers, but you need to be a little advanced user of Linux, so Linux plus some basic programming techniques. Yeah. So if we are good enough to go, can we say today is a day? And yeah, one more question: from where we can get more knowledge on this topic? Okay. So I would like to know, Pratik. Pratik, are you an undergraduate student or postgraduate student, or are you doing your own research? And what levels of knowledge you would like to learn more? So in case if you are an undergraduate students, you need to learn the BTech bioinformatics. Okay, okay, it's great to know, Pratik. I would like to give my email address also. Just write to me if you are a bioinformatics students, right? We have internship programs, so yeah, in LinkedIn, but you can, I don't remember the name. I mean, I don't use LinkedIn often, but I use the email. So you can write to me. So at the same time, you can get more internships or research projects or any other stuff here. To learn more bioinformatics, it's not, I mean, even if you spend 5 years, you can't. I mean, no one in this world can learn everything in bioinformatics. If you ask me, I might know around 0.0001 percentage of total bioinformatics knowledge. I mean, because it's very huge. There are artificial intelligence and deep learning and so much on, right? So we might be talking about very basic stuffs. Okay, so it's very hard to learn everything in this world. But if you want to have a particular idea on research projects. For example, whole genomes or whole exomes or whole metagenomics sequence analysis, we can help you on basic trainings. Okay, and there are basic algorithms, statistics, and basic knowledge of very basic programming and very basic Linux system, text user interface usage. And then you will be good enough to go ahead with the analysis. And of course, you need to know basic molecular biology and biotechnology to start and go ahead with this. Okay, so thank you so much for everyone for patiently waiting for the session. And I know the session was too rush, but if anyone wants to contact me later, you can contact me. Then, yeah, and thank you so much for Blooming Minds online classes for inviting us to do the basic session. And there was one more question also. Do you offer online internship? Yes, we do offer online paid and not paid internship depending on the students and their research project. Most of them are paid online internships, but we can. It depends on the research projects and the students' willingness. So you can always contact us. Then link for internship. Actually, I don't have the. I think can help you by providing internship, but anyway, you can write to me, right? Right, write to me on this email address about all the information. Yes, and Dr. Somalia Das. Okay, yeah. Thank you so much. Okay, so over to you, Blooming Minds online classes. Yeah, thank you.